Good afternoon, everybody. And I see that we've got some of our scientists from R&D that have connected. So good morning to you as well from the USA. So yeah, thank you very much for inviting us to present during this session. I think it's very interesting to have these types of presentations. I remember back when I was doing my PhD, I would love to learn about the different possibilities outside of the academic sector to see what is possible to do in certain countries. I was looking into France in particular. So I think it's great to be able to share feedbacks from people that have been in your shoes and to give some experience of what we've done, where we've been, and where we could potentially be going in the future. So from my side, I will be discussing from the lab to market strategy, and then afterwards Samuel will be talking about from lab to sales channels and sales management. And afterwards we have Benjamin Roussel, who will talk about tech support, and then two scientists from our R&D in the US, who will talk about their work there as well. So can you see my screen? Is it working well?
Yes, fine.
Great, fantastic. So quickly, a little bit about myself. As you can see, my first name is Matthieu, which is a French first name, but my surname is Lewis, so very much British. I was born and raised in London, in the UK. I chose this picture because it looks a lot like London, but the weather is never like this in London. So it's a good opportunity to say that, yeah, it is sunny sometimes, but not mainly. And then my mother is French and my dad's English. So we moved to France when I was a teenager. We moved to the lovely city in the southwest of France called Bordeaux. And here we have a quick snapshot of this great city as well in France. During my studies, so I went on, so when we moved to France, I just did the regular high school diploma in France, and then went to university at the University of Bordeaux. And one of the fantastic opportunities that I had was to spend 1 year abroad at the University of California, Santa Barbara. So this was during my undergraduate degree. So I did an undergraduate degree in biology. And as you can see here, so this is a picture of the campus. It was very nice. It was next to mountains and then next to the beach. So we had private beaches for the university. And yeah, it was a very, very nice campus. And during that year, I really started to work a little bit more on human biology, human physiology, and really focusing a lot of my time working on genetics. So afterwards, I came back to Bordeaux and did my PhD in genetics at the University of Bordeaux. And I understood quite early on that I wanted to go into the industry, the industrial sector straight after my PhD. So I applied to work at Takara Bio in Paris. And here is a picture of Saint-Germain-en-Laye. So the city that Takara Bio, so the European headquarters, is based. So it's based here. It's in the northwest of Paris. And we see Paris in the background and we see the clouds. So it's very cloudy, apart from today. So today we have a very nice day. So there's a little bit about like my pathway, traveling a little bit, studies in Bordeaux, a little bit in the US, and then now based in Paris. So, I mean, as you know, when you go on to do master's degrees, PhDs, and then postdocs, there's a lot of traveling that can be involved as well. And it's fantastic to be able to see these different cultures and different cities as well. So in April 2019, I finished my PhD. So my PhD was working on CRISPR-Cas9 technology. So some of you may know of this technology. It's a tool that can be used in genetics to be able to modify genes in living cells, so eukaryotic cells. And back when I started my PhD in 2015, it was at the beginning of using these techniques to do genome-wide screening assays. And CRISPR was really starting to become more and more popular. More and more labs were using this technique, but it was relatively new. So CRISPR was discovered in 2012. And then from then, the technologies kind of just grew and grew. And the goal of my PhD was to use a CRISPR library. So it was quite technological. So we can kind of go into what this was, but to use it in order to better understand how certain patients that have leukemia, how they respond to treatment. So the lab that I was working in, it was working with scientists and with doctors who were in frequent contact with patients that had chronic myeloid leukemia. So it's a form of blood cancer. And there is a treatment that exists for this cancer. Some of you may be working on this as well. And this is great because there's a treatment that can treat chronic myeloid leukemia. However, for a subset of patients, they are resistant to the treatment and the treatment doesn't do anything for them. So the goal of my PhD was to understand which genes could be involved in this resistance and if we could identify any ways to bypass this resistance to optimize certain treatment protocols for patients. So during my PhD, I was in 3 different scientific publications. 2 of them were first author publications. And what I really enjoyed was the communications aspect. So going to many different congresses in France, but also around Europe and in the US, and talking about this work and talking to other scientists that were in this domain and sharing ideas and sharing protocols and technologies. So to resume my PhD in one slide, I decided to kind of do a graphical abstract that I took from back in the years when I was in the lab. So basically the goal of my work was to work on cell lines. So there were specific cell lines for chronic myeloid leukemia. I then applied a CRISPR library to these cell lines, which means that every single cell in the cell culture had a different gene that was knocked out. So here, every color can kind of be a symbol for a different gene that's knocked out. Then what we did is that we would apply imatinib selection. So imatinib is the drug that's used to treat chronic myeloid leukemia. And what we saw here is that the majority of the cells died. This is good. It means that the treatment worked and that we killed the cells. However, there was a subset of the population that did not die. So it means that these cells were resistant to the treatment. And the goal of my PhD was to understand which of the genes were involved in this resistance and if there are ways that we can bypass this resistance to offer certain treatment plans. So we used CRISPR technology, many molecular biology tools. We used NGS, some bioinformatic softwares, such as this one called MAGIC. And we had identified around 50 different genes that were potentially involved in imatinib resistance. From this, you can find many kind of well-known tumor suppressor genes. So we were really looking at these different genes to see if they could be potential druggable targets for treatment. Then what we did is that we had validated, so we organized these genes according to the FDR score. So it's the false discovery rate score. And then we wanted to better understand if all of these genes had a specific resistance individually. So we generated individual cell lines with knockouts in all of these different genes and studied these cell lines individually to better understand the mechanisms that were underlying the resistance to treatment. And from this, we saw that there were 2 main pathways that were affected, the apoptosis pathway and the MAP kinase pathway. So what we then did is that from this, we did some treatment combinations where we would use imatinib plus nabitaclax, which is a drug that can kind of activate apoptosis. and then imatinib plus pimasertib, which is an inhibitor of the MEK pathway. And then with these combinations, we saw that there was a way that we could recover sensitivity, and the patients that show resistance could potentially use these two drugs, and the treatment would work better for them. So this was the work that I'd done during my PhD. There was a lot of collaborations with doctors and scientists around Europe to better understand the roles of these genes, and yeah, so it was a good way that we could move forward, not only better understand chronic myeloid leukemia, but also better understand the use of CRISPR-Cas9 and these different screening approaches. But also during my PhD, what I really loved to do was to communicate about science. I thought that it was as important as the actual work is to get the message out there. And in France, there is a national competition called MT-180. So in French, that's Ma thèse en 180 secondes. So in English, it would be 3-minute thesis. I'm not sure if it exists in Austria, but there may be an alternative. And basically, it's a national competition where PhD scientists, so young scientists, can present their work in 3 minutes. And it's meant to be for the greater public. So you're basically supposed to present your work to people that aren't experts in just life science in general, that really maybe don't know what DNA is, and let alone know what CRISPR-Cas9 technology is. So the goal here is to do these types of presentations, and I really enjoyed this. So I think that if there's a possibility, if you're interested in going into the private sector, interested in working more on communication aspects, to better talk about your PhD and to talk about the science, look to see if there are these types of events that are possible in Austria. You learn a lot from these events, and then you potentially can present this work in front of hundreds of people. So it's a very good opportunity. With this opportunity, so we were actually invited to talk on some national French radios, so France Inter. So that was quite fun. And also the finalists of this competition, of which I was in. So we had some certain days where we would use the Twitter account of 3-minute thesis and kind of present what it's like to work in a lab. So an everyday, like a day in my life on Twitter. So that was really fun, learning a little bit more about communications. On top of that, there was a second communications festival that we really enjoyed, which was called Pint of Science. And I think Pint of Science is in Austria as well. So basically, this is a 3 or 4 day festival. It's once a year. And basically, this is a collaboration between the company Pint of Science and scientists around Europe, where they can present their scientific work in pubs. So it's really fun. Lots of people come, they get a beer, and then they learn about science. So here we did a presentation. I think this was during the last year of my PhD, or the second last year of my PhD. We started to talk about people with beers, and we started to talk to them about CRISPR technology, gene editing, and all of the consequences that gene editing may have. So interesting as well to go into these discussions. But also, as I said previously, it's not all about just communicating about just CRISPR-Cas9 and basic science in general. There was also a lot of communication in certain schools. So the European School of Hematology, the French School of Hematology, the American Society for Hematology, talking to doctors and seeing really how the certain research can potentially impact their work. So communication for me was important during my PhD. So as you can see on the left-hand side, these are kind of the technical skills that I had acquired during my PhD. CRISPR, there was some NGS, lots of molecular biology, so really wet lab work. There was a focus on cancer research, so on oncology, but also quite a bit of scientific communication to experts, but also to the general public. And near the end of my PhD, there was a job offer at Takara Bio Europe. So I checked out this job offer and I saw that TBE, so Takara Bio Europe, was based in France. So I was interested in staying in France. And a lot of their technologies, so I'd used some Takara kits when I was in the lab, and some of you may be using Takara kits as well. And a lot of their technologies were based on stuff that I'd done during my PhD. So NGS, lots of molecular biology, cloning, cell culture, and also CRISPR-Cas9. So I decided to apply for this job, and the role that I had at the beginning was a tech support specialist and product manager for PCR and for RT-qPCR. So really stepping away from wet lab work, R&D, and going more into technical support and into the marketing team. So I won't go into details into the role as a tech support specialist because Benjamin's here and Benjamin will be able to explain to you what his life as a tech support specialist. But I can talk a little bit more about the marketing aspect and the work that we had initially as a product manager and how that evolved over the years. So when I started here, I was quite naive in regards to marketing in general. I joined the marketing team. I knew that there was a big learning curve. Technologically, I understood the technologies and the products, and the tech support was great to talk to scientists about this. But for the marketing aspect, there was a lot to learn because we didn't really have that many marketing courses during our PhD in Bordeaux. So initially, when you think about marketing, you think about adverts, you think about maybe communications as well. But in fact, it is much more complex than that. Here's a brief resume of kind of all of the different aspects that you can have when working in a marketing role. There's what we call upstream and downstream marketing. Upstream marketing is really kind of better understanding the market, understanding what are the developments that we may have and what can we produce for future applications. And downstream marketing is more the marketing that you're maybe more used to seeing, which would be communications, certain adverts and everything. And in between, there is a myriad of different possibilities and different focuses that you can have. Like, for example, before a company such as Takara Bio generates a certain kit, it's because there's been intensive strategy and marketing beforehand. They analyze the markets, they analyze the competitors, they can analyze what the customer needs, how we can offer something that's slightly different, how we can be more competitive in regards to the competition. So there's really a lot of work here. And thankfully, during my role as a product manager initially, and then going towards market strategy, you learn a lot. So you learn a lot as you talk with the team. So obviously, we have a manager for marketing. We are different product managers, different market strategy managers with more or less experience, and you continue to learn from this. And you can go through certain courses to better understand market strategy. So it's really, really interesting. Marketing is much more than the adverts and some communications that you may see as a scientist. It's really the whole aspect of the development of certain products and how these products are communicated into certain markets. So a case study, an example of this, and kind of the roles that we had and the roles that I had as a product manager was also, you can predefine certain aspects. You can work on developing certain products for 2 years, for 3 years by analyzing the market. But sometimes life happens and you have to react very quickly. And you need to work with a team in order to develop an efficient message and in order to get to a certain customer. So, as you all know, in the early days of the COVID-19 outbreak, testing was increasing. PCR testing was increasing. There were certain protocols that had been developed by scientists in Berlin, scientists at the Pasteur Institute, scientists at the CDC in the USA. And all of these protocols included RNA extraction. But RNA extraction requires machines, RNA extraction requires a lot of plastics. And what had happened is that during the months of February, March, and April in Europe, when the pandemic really started to take off, all of the RNA extraction solutions had been used previously in Asia, because in Asia, in the earlier months, so December, January, and February, they started to test a lot for COVID-19. And all of the plastics that were generated by many companies around the world, they were all used for these initial tests. So when the European pandemic started and when the US pandemic started as well, so these are the markets that we're working with, there wasn't a possibility to work with RNA, with extracted RNA. So we would talk to certain hospitals and they'll be telling us that they didn't have extraction solutions. So they were doing some homebrew techniques to be able to boil the virus and then use that for RT-qPCR, but it wasn't working very well. And they were really looking for alternative protocols. So what we did, and this is one of the marketing activities that we had predefined, and then we said, okay, we need to work on this to try and help as many scientists as possible to develop new protocols, is that we previously had a kit, so a one-step RT-qPCR kit, so a research kit that can be used directly on on samples. So there's no need for RNA extraction, but our R&D, because this kit had been developed way before the pandemic, our R&D hadn't validated it on viral transport media, so the media that you have when you have the COVID test with the nasopharyngeal swab samples. So what we decided to do is to work with hospitals and labs around Europe by supplying some of these enzymes for free and saying, okay, we can give you a kit for, let's say, 200 reactions. You can help optimize certain protocols, and then we can share these protocols within this network so that when certain labs need to perform COVID testing by RT-qPCR and they don't have RNA extraction solutions, they can potentially adopt this protocol. This was a very nice strategy and it worked. So we started talking to many different virology labs. We were working together to develop certain protocols. This is what we call a door opener message. So when we would send them this message saying we can collaborate with you, the scientists and the clinicians were very interested in discussing with us. They would also look at the different solutions that we had in our portfolio. And afterwards, what had happened so very quickly is that we had worked with these 50 odd different virology centers around Europe. We worked on certain guidelines, so certain protocols that can be potentially shared for direct COVID testing without RNA extraction. And from that, because we're working with many scientists, there were many publications that were then released looking at either a direct protocol or alternative protocols, or just looking at different solutions and optimizing the COVID-19 testing strategies. So it was a very, very rich period where we started to talk to a lot of different customers, and we were really sharing this information to try and get COVID testing more efficient, less reliant on RNA extraction, and really to continue to develop this. So publications that we could share, there were certain guidelines that we could share. And also because we have an R&D and we can produce products ourselves, we were also working on solutions to optimize this form of testing. So more complete solutions for testing that can be used with saliva samples, for example, or different types of samples. So we're continuously working with the market, and we're continuously working to optimize our offering when talking to scientists around Europe, the Middle East, Asia, or the US mainly. So how did my role kind of evolve? And this is something that you can see in the private sector, in the industrial sector, is that your role can change very quickly. So I had started in 2019 as a tech support specialist and a product manager. This was really the period of lots of learning. So learning a lot about product management, about marketing, lots of communications when talking to certain customers, and then internal communications with the team. It was a transition period as well, going into the industrial sector. In 2020, I became just a product manager. So I didn't have the tech support role anymore. I was really focusing on the management of certain products in our portfolio, with really a heavy focus on COVID-19. And during this period, you needed to have some form of flexibility. We needed to be able to talk to a certain lab in Denmark, and then quickly switch to another lab in the south of France and in the UK, and then have this type of flexibility to quickly be able to modify the guidelines, to share the information, and to collaborate with a maximum of people at the same time, going from one subject to another. In 2021, this is when my role evolved yet again. So from going from product management to market strategy management, where here we really have to better understand what are the needs for the longer term future. So when we look at currently what's going on for molecular diagnostic testing, the idea is to say, okay, what's going to happen in 3 years? What's going to happen in 5 years? And how can we work with scientists and with our R&D to develop the most optimal tools for that time period? So really here, my role is focusing on molecular diagnostics and what we can develop in the future for in vitro diagnostics in particular. So this is when you learn a lot more. You learn really a lot about marketing strategy and how this can really impact a company as a whole, and how your role can potentially evolve. And in 2022, I had the role of senior market strategy manager. We had recruited a product manager that works with me for the molecular diagnostics team. And we really have lots of collaborations with our affiliates, whether they be in Japan or in the USA. So we're continuously talking to more strategy managers, to certain product managers as well, and also importantly to our R&D, who you'll talk to later in this session. So very, very interesting last 3 years. I thought this was a great transition from the academic sector to the industrial sector. And I'll be free to take any questions you may have, and finishing with the slide where we finish by saying that's good science, because we have these solutions that can help for better science. So thanks very much.
Thank you very much. This was a great talk. I got some questions in the chat. They wrote me directly. When you would describe your daily tasks for your working day in 1 or 2 sentences to get an idea.
Great, yeah, sure. So daily tasks, sometimes you prepare for certain daily tasks, and then you have to be flexible because things can change very quickly. So typically, what would happen is that this is something that changes a little bit from the academic sector, is that the companies, well, at least Takara Bio Europe, we're more of a larger company. There's a lot of communications that needs to be done internally. So we have meetings, we have more regular meetings that I used to have during my academic work, where we have daily meetings with the marketing team. This is really kind of to kick the day off. We talk about kind of everyday subjects, but we can also quickly go into what needs to be done during that day. We can give certain tasks. And then we work on, really either talking to scientists that are working. So for me, scientists that are working with molecular diagnostics, we spend quite a lot of our time as well talking to our current customers. So our customers are scientists, but our customers are also biotech companies, quite large companies that develop molecular diagnostic assays. So we need to maintain a good relationship with them, and we need to tell them about all of the news that we may have and the different ways they can optimize their protocols. And then we have certain longer term projects. So really, when we're talking about the market strategy aspect and all of the different points in blue that I had shown for product management, that is a lot of work. Analyzing the markets, analyzing what are the expectations from the markets, and then sharing this information with our R&D, preparing presentations to be able to maybe convince our R&D that we need to go in a certain direction or not. So it's a lot of office work. So there's not a lot of wet lab. We do travel, so we go to conferences, we visit customers when we can start visiting more customers. So it's starting up again. So it's very, very diverse. And this is where you need to have some form of flexibility because you can quickly have one focus, and then all of a sudden there's a new priority that you have to deal with, and you have to kind of deal with it quickly. So be agile.
Be agile. Okay, thank you very much. How big is your team you're working with, and how is the decision making done in your company?
Yeah. So currently the marketing team, so we have a team that's split in two, basically. So there's one part of the marketing team that's more product management and market strategy management. So this is a team of scientists and also a pharmacist. So really everyone has a PhD. Most of the MSMs, so the market strategy managers, have also done postdocs. And pharmacists. So for us, this is a team of 5 or 6, and it's continuously growing. And then we also have the second part of our team, which is more of the downstream marketing. So this is these are people that have got degrees in marketing, so not scientists, and we really work with them in order to collaborate for all of the different aspects of marketing. And there are 3 or 4 people in that team. Mm-hmm. And then we have a head of marketing who helps us a lot and can guide us through all of the marketing aspects. For the decisions, so we take some decisions on the initial decisions on a scientific aspect. We develop certain propositions that we would then present to upper management or to our affiliates abroad. And then the way that we present this and the way that this information has been backed up by facts and by data that we've collected, it can then influence the decision that's ultimately taken by upper management. But our role is quite significant in choosing kind of which way we're going to go. So we do have quite a good role in regards to the vision of the company and how it can progress in the future.
Mm-hmm. Thank you. One last question before we have to move on. You said that you also have postdocs in your team.
Yeah.
What's the general time there were postdocs in academia before they join your team?
Yeah, so it really depends. So I know that there's one postdoc who spent 7 years in Italy. So he did 2 different postdocs in Italy for 7 years, and then joined Takara as tech support, product management, and then evolved as well to senior market strategy manager. There is another postdoc who had done his postdoc in New York. Another postdoc had done her postdoc in Paris. So it really depends. There are people that have done up to 7 years postdoc, and then I haven't done a postdoc. So it was just directly after my PhD. So it really depends on the motivation of the candidates, and it depends on kind of the experience that they obtained during their PhD.
Mm-hmm. I think these other questions we can maybe then spare for the second session. Anyway, you decided to stay a little bit longer, Matthieu. I will hand over now to your colleague.
Yeah. So Sam, you're welcome.
Sum it up. It's very interesting that especially this postdocs, PhD. Also, you can have a PhD and then you can make a postdoc and you can still then enter industry. I think this is also a viable option for a lot of people, and we should keep that in mind, that it's nothing. There are no lost years, so to say. You just have to find your fit.
Exactly.
Thank you very much.